when I started my lab, one of the first things that we've done was like I was sort of like tired of reading those papers after reading all those 1,000 papers. So there were a lot of databases that came up that had a kinase substrate information from papers. So those databases basically have a name of a kinase and then all of its substrates, and that was the database. And there were 6 databases that had that information where people went to papers and then they extracted that kinase substrate relationship. So we merge all those 6 papers and then we integrate them and then create this tool that allow you to put in a list of proteins and then prioritize which are the most relevant kinases based on enrichment analysis. And we've done it also for ChIP-seq and transcription factors. At the same, right after we published that kinase enrichment analysis paper, published this other tool called CHIA, which is ChIP enrichment analysis. So we did it with Kia and Chia, but in reality, there is a whole lot of big data sets out there that could be converted into the simplified format that I mentioned, that gene set library format. And these are only some of the icons that represent those resources that you can take and convert them to this simplified format where you have some type of a label and then you have a gene set that comes off as a part of the label. Okay. You can also do it for other things, so it doesn't have to be genes, but genes are really central in this type of approach. So what we realize is that now we can basically go through this process of converting resources into these simplified data structures, and I'll show you later why it potentially can be useful. So this is a little bit more of a drill down to this idea of you can take all of those resources, for example, the cancer genome atlas, TCGA. If anyone heard about TCGA? Okay, some nods. So basically, it's a massive effort by NIH to follow and collect data from cancer patients and their tumors. So every tumor is sequenced, and then also microarray and RNA-seq are applied on the tumor, and proteomics, and also the patient data is there where have their diagnosis and survival and other what the drugs they were on, stuff like that. So this is done for many cancers, and this type of effort is going to grow to much greater in the near future. You'll have massive data sets. So in this particular case, you have data from tumors, and those tumors are connected to their mutations. So there is that data is in here. There's other data in TCGA that could be converted, but you can create a bipartite graph that connect tumors to mutations. And then those tumors, they come from different tissues, and then like for this, like the tumors, you can also part some of them, or they could be associated cancer cell lines that are used to either take the tumor and convert it to a cell line, or find ways to see which cell line is most similar to a specific tumor, for example, from TCGA. And so you can create a correlation, the matrix between, let's say, hundreds of cell lines and thousands of tumors. So the idea is that you can have a bipartite graph for that, and then you can treat those cell lines with drugs, and then see which drugs are, let's say, inhibit the cell lines, the viability. So this is done with CCLE and CTV square, which are two projects, NIH projects, that basically try to they have a massive list of cell lines. For CCLE, it's over 1,000 cancer cell lines, and they treat those cell lines with many drugs. And then they have viability data. So now you can also go like from the drugs directly to the tumors, because you also have that relationship. So this sort of like diagram can give you a whole full way to potentially go from, let's say, diseases, like human diseases, to genes that are knocked down, have mouse phenotypes. So all of those things can be potentially connected as you start simplifying and abstracting this map. So this is what I was saying, like those resources, in general, they fall into 7 categories. So the first one is drug or gene knockdown followed by genome-wide expression. So the project, like this data is in Geo, but also is a part of LINKS. Anyone heard of LINKS before? The LINKS program? I guess you know about it, because I think you were worked on it while you were in the lab, or not, but you heard about it. So LINKS, basically, the idea is that you have a bunch of human cells, mostly cancer cell lines, but other cells, and then you treat them with a bunch of drugs, measure the gene expression before and after drug treatment, but also proteomics, cell viability, and this is done across multiple centers. So this is just one type of data. You can find a lot of those studies in Geo as well. Epigenomics are transcription factors, the ChIP-seq type of studies, as well as other epigenetics modification studies. That's the stuff that I showed you, the antibody. And this is the drug and knockdown effect on cell viability. So I mentioned how those consortia, they use a bunch of drugs to see how drugs affect viability of a bunch of cell lines, but also knocking out each individual gene in those cells and seeing how those cells survive. So this is another type of data, and then only MGI, they're looking at mutations or knockout of gene and how it's connected to a phenotype. So now you have a bunch of human or mouse phenotypes, and then you can connect those phenotypes. A dictionary of phenotypes can be connected to a single gene based on whether it's mutated or knocked out. And then this is the collection of expression for patients or tissues from normal patients, and this is the GTEx project. It's an important project where they take post-mortem tissues from people that volunteer their organs to science, and then they do gene expression on each of those tissues as well as their genome, sequencing their genome, and they're going to have about 10,000 of those patients right now. You can get access to about 500 of those or 800 patients and their expression in each of those organs. BioGrid, okay, and this NERSA project, anyone can, I guess it says that, but protein-protein interactions, pathways, metabolic pathways, and cell signaling pathways, those are physical interactions between proteins and other cell signaling molecules. And then these resources connect drugs and small molecules to phenotypes.